Hello, good morning.
Good morning. Hi.
Morning.
Hey. Okay, our speaker just texted. He's logging in now. Hello, good morning.
Good morning. Hi.
Morning.
Great. It's going to be the last Neuroccino for 2021. Can you believe it? It's another year online meetings done. Good morning, Abby.
Hello. Good
It
morning.
looks like it's snow outside your window.
No, it's not so. I should be fun, but not yet.
Hang on. Sorry, Thomas can't find the link. So I'm just sharing it again. Tuck. Here.
Okay.
Morning, Valentina.
Good morning, everyone.
Right, let me share the paper already that he's going to present. Here we go. Looking good. There he is. Good morning.
Hello, good morning, everyone.
Good morning.
Right. So here's the link for everyone, and I'm going to share it with everyone over on YouTube. Great. Now let's make a start. It's 9:31. Welcome to Neuroccino, our weekly online journal club, where everyone is welcome to present a paper they came across and thought it was interesting, or a book chapter is really up to you. Anything that is sciencey and takes your fancy, we can discuss here. Today we have Thomas, who's a PhD student at ICM in Paris and currently visiting our lab in Bordeaux. And he chose a paper called Correlates of Anosognosia in Alzheimer's disease and Mild Cognitive Impairment. Now, Thomas, if you're ready,
Yes.
just share your screen.
Okay, sure. Okay.
And tell us why you thought this paper was interesting.
So thank you for the proposal to present today. Well, where I find this article when I did my bibliography on the multimodal approach of anosognosia. And what I find interesting here in this article, and we will see why, is because they study different measures of anosognosia in very different ways. So there is a self-evaluation questionnaire, which is very standard, some neuropsychological approach, and VBM correlation, so for voxel-based morphometry. So this is why it is interesting, because normally a lot of papers do only one of the three approaches, but not the three in the same time. So that's why I wanted to discuss this article with you. And maybe also because a lot of projects in the Gene Lab is related to those methods. So I think it would be interesting to discuss it with you. There is only one point that we will discuss after is the small samples here of this article, because there is only 27 patients, 15 NCI and 12 AD. So there is very small samples to do some VBM analysis, but we will see why it can be still interesting. So what if we go through the papers, we can see that the three methods to measure anosognosia are really standard, because there is a clinician rating which has been seen in the literature. The second way is the patient-informant discrepancy scores. What we can say about it is that these first two methods are very standard, but the third one, which is the performance score, based on the self-evaluation of the patients on neuropsychological tests, is a bit different, and there is no gold standard in the literature to measure anosognosia with this way. And this can be very interesting because based on which test and neuropsychological test you will do with the patients, you can have different scores. But here we will see that it can be also an issue because in this particular paper they focused on executive function and episodic memory functions. However, the proportion of tests between executive function and memory functions is up to the authors, so you can have a lot of discrepancies in the literature because it's up to the authors and there is no gold standards. And maybe to finish the introduction with the last point, and that's why I choose this article, is because based on the literature, there is no studies who analyze in the same time the three different methods, and so the self evaluations, neuropsychological evaluations, and the clinical evaluation of anosognosia. And they did. This is the first study who did correlations with VBM analysis. So concerning the materials and methods, here is maybe the biggest limitation of the study, because there is only 27 participants in this study. We will see that a lot of results in the discussion are related to previous findings, and it goes in the same way than previous findings. So we may say that some of the analysis correlates with other papers. So this is a good point for this article, but still it can be tricky to do strong statistical analysis with only 12 AD patients. Here are for the neuropsychologists, here are the tests used for the correlation. So there is again no gold standard for neuropsychological assessments of Alzheimer's, but still those tests are focused on the memory and executive function, which are the main function altered in Alzheimer's disease. So this is concomitant with the literature. And what to say about the Anosognosia Questionnaire? Well, as I said, maybe that's here we can see that when patients are asked to rate themselves on their performance on neuropsychological tests, they can say that from 0 to 4 points for each test. And there may be one issue here also, because some of the tests are really focused on one specific items or one specific cognitive function. For example, if you take the Stroop test, it will be a very strong executive test. But if you take the frontal assessment battery, there is 6 subscales in this test. So when they have to rate themselves, it will be tricky for patients because they have to perform 6 different tests before giving them a score. So it can alter the score. I'm not an expert on VBM, so I cannot say if the pre-processing steps are the best or not. I didn't discuss about this article with Michel, but it looks like the same steps that I did with him during the last 2 weeks. So I will say that I was not surprised with the steps, but that's all what I can say. And there is one point that I find maybe it was interesting, but it was not maybe not the best idea here. In that they had a work group of 27 participants, patient MCI and AD, and they decided at the end to perform the same analysis, not on the 27 participants, but on the 2 subgroups. So they did statistical analysis on 12 patients. And we know that correlation analysis on 12 with 12 samples can lead to to statistical issues. If we go through the results, I think that the best idea is to check at the tables, and I will give a small resume of what they did. So first, we can say that here in the first table on a demographic and neuropsychological test, the only significant difference after correction is on the MMSE, which is a standard neuropsychological test to assess dementia. So here there is no much to say that AD patient has more dementia than MCI group, but there is no difference on all other neuropsychological tests, even on the IQD, which is a questionnaire to assess the dementia, so to assess anosognosia. And we can see that there is no difference of anosognosia, significant difference of anosognosia between our 2 subgroups after correction, which can be like interesting, because it could mean that who are the groups may be at the early stage of the disease, because we know that anosognosia in Alzheimer's arrives after several months, so during the last steps of the disease. If we check now at the correlation matrix between self-questionnaire, self-evaluations of anosognosia and all the neuropsychological tests, we can focus on the 2 first rows because there are the correlation between the questionnaires and this is the correlation between the 3 different methods, and we can see that there is only correlations between the anosognosia scales, so the IQD, and the frontal assessment battery, meaning that the anosognosia seems to be correlated with executive function mainly, and not with episodic memory functions. This is interesting because in many in the first steps of the literature on anosognosia in the early 90s, for example, Michon et al. Said that anosognosia was mainly focused on frontal lobes and frontal areas, and it's only since like the last 15 years that we have first correlations between temporal structures and so episodic memory and anosognosia. So this first table seems to correlate more frontal areas with anosognosia, less than temporal regions. If we go now, if we see here in the table 3, the first results of the VBM correlation, so between the different neuropsychological tests and questionnaires with the structures of temporal and frontal structures. I put in green the hippocampus because this is the main regions which correlates between all the tests and the damage in the structures. So it seems that the more the patient is anosognosic, and the more the hippocampus is altered too. This is very interesting because commonly in the literature, the authors said it in the discussions. It is always the hippocampus are altered in the early stage of Alzheimer's disease, but we can see that here in the first part that the hippocampus is also correlated with the evolution of anosognosia and with the self-evaluation of anosognosia. So this result is very interesting because it's one of the first who present it. And we will based on the discussion, the authors are looking for different structures in the brain. So if we go first on figure 1 and 4, we can see that here in figure 1, they mixed the MCI and the AD group. We can see that the hippocampus is on the right side is the main altered structures, and there is a correlation between the alteration of the hippocampus and a poorer score in ADQ. So this is very interesting. If we go to table figure 4, we can see that they did the same analysis, but they separate the MCI and the AD group. And here, this is interesting because we find the same results on the hippocampus, but only for the MCI group, whereas for the AD patient groups, the correlation is more focused on the anterior cingulate cortex. And there is no, in the discussion, the authors did not really interpret these results and with the difference of evolution between MCI and AD. So one idea could be to maybe more focus on this part because maybe this is the most interesting results, meaning that with the evolution of the disease, the correlation moves from the hippocampus to the anterior cingulate cortex. So we should explain why, and this could be more discussed. In figure 2, it is really interesting because it is the results are correlated with the only significant correlation is between the Stroop between the executive function and correlation with the anterior cingulate cortex and anterior cingulate gyrus and also the frontal-orbital cortex. So meaning that based on the different cognitive task, for the executive function, you will have damage to the frontal lobe, and for the more memory function, you will have damage to the temporal lobe. And this is interesting because in the literature, as the author said at the beginning, we can have damage focus studies are focused only on the frontal lobes or focused only on the temporal lobes. And this is one of the first story who decided to focus both on temporal and frontal part. And we can see that damage can occur to correlations can occur to both structures, and maybe that anosognosia is not only related to damage to one structures, but can be anosognosia could be due to damage to many structures, and could be separated in different subscales. We can have anosognosia for an executive function, but also we can have anosognosia for temporal function that has episodic memory or autobiographic memory. And it is important to separate all these subscales. And the last part is very interesting because they decided to study the correlations between the 7th measure of anosognosia that I presented in this paper. So there is 7 subscales of anosognosia, and if and when the author tried to correlate with one global score all those 7 subscales with one particular regions, it also leads to a correlation with the hippocampus and with the right hippocampus. And the conclusion that the authors did is that it could mean that the key structures of anosognosia is not in the frontal lobe, but more in the temporal lobe in the hippocampus. So I don't know if it was too long or too short, but the last point that I'd like to discuss is this one maybe, and because I studied it in my PhD, so I'm not totally agree with the authors, because their main ideas is that based on the last result I show you on the hippocampus, the idea of this article is to show that maybe anosognosia is led by damage and alteration of the hippocampus, and that there is no, finally, there is no different subscales of anosognosia, but only a global leading process of anosognosia in MCI and AD, and that this global, this model is only based on the role of the temporal lobe and more precisely on the role of the hippocampus. So I was really happy when I started reading this article, because at the beginning I was, okay, this is the first article who not only focus on the temporal lobe or not only on the frontal lobe, but at the end, the conclusion is that frontal lobe, there is no conclusion concerning the frontal lobe, but they decided to focus only on the temporal lobe. So this is a conclusion, but I think that they could have interpret all alteration to different structures, such as orbitofrontal cortex and hippocampus.
That's it.
Yeah, it's yeah. Of course, the last there is the last limitation, and they discuss about it in the last part of the article, is the sample size of patients, because of course 12 patients is really, it's really not much to have a stronger correlation.
Lovely, thank you. And we can discuss that in a minute a bit more. Just to reiterate, for those over on YouTube watching this debate, if you have questions, just write them in the chat and I will bring them over here to ask Thomas. Wow, we already have the first question. Valentina, take it away.
Thank you, Thomas, for presenting this interesting paper. Big question. I was wondering, did they control for the memory impairments in their analysis, the VBM analysis? Because we are talking about patients who have memory problems, and they're pointing to the hippocampus as correlated with anosognosia. And basically the measurement was asking the patient to rate their own performance and then compare their rating with the rating of the caregiver or clinician, I don't remember. So could it be related to their memory problems, the different rating, and therefore the VBM analysis being linked to these memory problems if they did not control for it? Maybe they did.
Yes, this is an interesting question. And so what was interesting is that they did both. So there was the part of a discrepancy between the caregiver score and the patient score. So that was the first part. And they did also the same with between the clinical scales with the physicist and between the patient and the physicist. So the physician and the caregiver did both an evaluation of the patient performances. And concerning more precisely about your questions, if we see here in the table 2, is that both self-evaluations of the performance on neuropsychological tests and the psychological evaluations themselves correlates. So it seems that the objective way of the evaluation process for neuropsychological items goes in the same way than the way the subjects evaluate themselves. But it's mostly happens on executive function and not on memory function. So yes, maybe there is no strong correlation on memory functions because there are damage to memory to the hippocampus and other memory related structures. So yeah, but they did not do additional checkings.
Okay, thank you.
MV has a question for you as well.
Yes. Thank you, Thomas. So yes, so I checked a little bit the paper. So yes, one of the limitation is for sure the small sample of subjects. But another thing is I'm kind of concerned about when you have two groups and you start to do correlation and say, okay, I'm mixing the two groups together. For me, it's always a red flag because you don't know when you are looking at your correlation if it is a difference, not a difference, but a correlation, or a difference between the two groups. And in this case, it seems to be very difficult to distinguish between the correlation and the group differences because they are looking at MCI and Alzheimer disease patients, and they know the test, all the tests are different between these two groups, and after they do correlation with this test. So you don't know if it is due to the group difference or the correlation. So they try to compensate that. So I looked in the paper, putting the MMSE in the correlation as a covariate of non-interest. But I would have been more interested if they put the group as the covariate of non-interest to see if all the correlation disappear after that. So I don't know if you have a guess or an idea on that.
Yeah, well, my guess on that is I totally agree with you, but at the end they tried to replicate the results they obtained on both groups doing the same analysis, separating AD and MCI, and that's why I was concerned about the fact that there were small samples, but they find the exact same for one part of the results. They find the exact same results when they separate the same groups. For example, to the localization of the altered structures, and on the last figures they show some differences, and for the with the evolution of the correlation from the hippocampus to the anterior cingulate cortex, and that's why I point out this fact in my explanation, because I was concerned about the fact that it's not the same structures alter. There is not the same correlation for the VVM between MCI and AD, but they did not discuss about it. Whereas this is a key point because there is not the same results when you do the analysis with the 27 participants or when you use the subgroups. So yes, it was maybe the most tricky point, and they should have more discussed about it.
Yeah, so I didn't. Yeah, so I was checking when you were discussing that. So, but yeah, but the figure 4 is yeah, it's uncorrected because they have a very small samples when they separate the two groups. So yeah, they try to manage that. But well, okay, thank you.
And Tina's coming back with another question, and then I have a couple of questions from YouTube for you.
Okay.
A more clinical question. So as you were saying that there are no golden standards for this kind of assessment in anosognosia for AD, do they comment on the clinical point of view, their samples, their groups? What I mean is, do they disclose the diagnosis? Do they have anosognosia AD patient or anosognosia MCI patient, and how many if they haven't read the paper?
Okay, sorry, I had a loss of connection. I did not hear the question.
Okay, it's okay. Do they comment if they clinically observe anosognosia for AD or anosognosia for MCI symptoms in their patients, and how many patients resulted to be anosognosic?
Well, they did not exactly comment the results. They did both analyses. They used the same scales. So what I know is that in the introduction part, they illustrate the fact that the questionnaire they use could be a gold standard because it has been proposed in different articles with a lot of more patients. They did there is an analysis with more than 100 patients. So they said that it could be a gold standard, but there is no more information concerning this part. Maybe because when I did my bibliography research at the beginning of my PhD, I found more than 8 scales to assess anosognosia. And even if there is different scales, they are all related to executive and memory functions. So I think it's depends how you write your questionnaires, but they all go in the same ways, meaning that they all focus on the same cognitive functions. So that's all that I can say.
Okay, so they don't say we have a thought patient that according to our clinical assessment resulted to be anosognosic.
They said just, that they consider a patient with anosognosia with a score of -2 on several items, but when they performed the group difference between MCI and AD, they didn't find any significant results. But they had for threshold some values based on different tests, but they did not comment it in the discussion. They just speak about it in the introduction part.
Great, thank you. Question from YouTube, which brings us back to the very first question, basically, that Valentina asked as well, which goes as follows. Good morning. Thanks for the presentation. I agree very much with your final critique. I would like to remind that the mini-mental state exam is a 30-point scale of which only 3 points evaluate episodic memory. Taking this into account, I agree with the first question. It is not enough to use MSEA as covariate of no interest, particularly with results that point to MTL as anatomic correlate. Aren't we measuring then secondary anosognosia to episodic memory problems?
Yeah, I totally agree with that. Especially when you find that in the correlation matrix, that's the most significant correlation is on executive function and not episodic memory. And this is a bit the point that I spoke about it at the beginning of the presentation when I said that because they didn't use a gold standard evaluation of Alzheimer's disease, I was a bit surprised that there was only few neuropsychological tests. Basically, when I work like in Salpêtrière in Paris, the neuropsychological evaluation is 3 hours evaluations with many tests on episodic and episodic memory, long-term memory, executive functions, and the correlations could be very more interesting because you can have statistical power with different tests, and I totally agree that one test is not enough to as covariate yet. Yes.
Well, especially the mini-mentals.
Yeah. Yes.
More than a house number, yes. Actually a diagnosis.
I think that the MMSE is not even like a strong neuropsychological test. It can be a first step to introduce subjects and to explain to the subject that there might be cognitive dysfunctions that needs to be assessed. But yes, it's not a global evaluation, a global neuropsychological evaluation, of course.
Exactly, yeah. Well, sadly, this is all we have time for. There's many more questions. We haven't even touched the anatomy yet, but we're out of time for today. So thank you so much for presenting, Thomas. This was the last Neuroccino for 2021, but we will be back in January next year. And in the meantime, while this is the last Neuroccino, we still have new content up on the channel. So we just uploaded the new Bordeaux T on the dead salmon fMRI study that you can go and have a look at. I just uploaded the Meet the Editor on registered reports with Chris Chambers. That is now up on the channel. And then next Monday, we will have a neuroanatomy and tractography workshop dedicated to the frontoparietal network and the arcuate frontotemporal network. So that's all available or coming your way within the next week. Thank you so much for joining us this morning. And do come back in January for more journal clubs. And as I mentioned at the very beginning, this is open to everyone. So anyone who wants to join and present a paper, you're more than welcome to do so. For those who only join us for Neuroccino, happy holidays and see you in the new year. Bye.
Thanks. Bye.
Bye.